This tutorial is for the AQA Biology specification, Unit 5, Chapter 10.4. We're going to be looking at the passage of an action potential in unmyelinated and myelinated neurons. So we're going to begin with an unmyelinated neuron. So as we know, the concentration of sodium ions is always higher on the outside of the axon membrane relative to the inside of the axon membrane, and the concentration of potassium ions is higher on the inside of the membrane than on the outside. So we can say that the outside of the axon membrane is positively charged and the inside of the axon membrane is negatively charged. Remember that these are relative. So the outside of the membrane is just more positively charged than the inside of the membrane. There's nothing to say that the inside of the membrane is negatively charged, it's just that the outside is more positively charged than the inside. So we just say that the outside is positive and the inside is negative. So at this stage the axon membrane is polarized. Okay, so what happens next is that a stimulus causes a sudden influx of sodium ions and causes a reversal in the charges on the axon membrane. This is called the action potential and the axon membrane is depolarized. So just imagine a stimulus hitting the axon membrane. What happens is these sodium ions start traveling through the axon membrane and the concentration of sodium ions within the axon membrane increases. So the inside of the axon membrane becomes positively charged and the outside of the axon membrane becomes negatively charged. And remember, these are relative again. So after the influx of the sodium ions, sodium voltage gated channels open further along the axon membrane. As the sodium ions are moving into the axon, they are depolarizing the axon, whilst those regions behind the new region of depolarization are closing the sodium channels and they are opening potassium channels. So this allows potassium to move out of the axon membrane and the concentration of potassium ions increases on the outside of the membrane relative to the inside of the membrane. What happens is so just stemming off that point, we just spoke about how the potassium ions are moving out of the axon membrane and the sodium ions are moving into the axon membrane. So we've got a higher concentration of potassium ions on the outside and a high concentration of sodium ions on the inside of the axon membrane. Now at this stage, the number of potassium ions that have moved out of the axon membrane is greater than the number that were originally outside the membrane. So there's been a really big increase in the number of potassium ions that are moving out compared to how many were there in the first place. So this part of action potential is called hyperpolarization. So this is where the outside is more positive relative to the inside compared to before. The axon membrane will return to its original charged state and we call this repolarization. So what happens is the sodium ions can go back to normal and can be actively transported out of the axon membrane, and the potassium ions on the inside, so we return back to our normal state, so resting potential once again. So it's sort of like a cycle. The neuron returns back to its resting potential, like I said, and it can be hit by a stimulus once again and the cycle will continue. We're now going to talk about the passage of action potential in a myelinated neuron. So just to quickly go over what a myelinated neuron is. A myelinated neuron is basically a neuron that is covered in myelin sheath, okay? And I'm just going to quickly talk about myelin sheath. Myelin sheath provides a covering to the axon membrane and is made up of several layers of Schwann cells, okay? You should have covered this in 10.2, which talks about the structure of a neuron and all these fancy words that I need to know the meaning of. So these membranes are made of lipids known as myelin, and you basically have neurons with myelin and neurons without myelin. So obviously we're talking about myelinated neurons now, but before we were talking about unmyelinated neurons. Okay, so we're just going to quickly talk about how action potential passes along these myelinated neurons. So the myelin sheath surrounds the axon membrane and acts as an electrical insulator. So as we know, action potential is an electrical thing. So when you have a myelinated neuron, in the places where myelin sheath is covering the axon, action potential cannot take place. So what happens is there are small intervals, or breaks if you like, in the myelin sheath that are referred to as the nodes of Ranvier, and the localized circuits arise between the nodes of Ranvier. Now the localized circuits are obviously just made out of the myelin sheath. So the action potentials will take place at the intervals of the nodes of Ranvier. So wherever you get those little gaps is where the action potentials jump from. So you get the action potential at one node and it will skip the myelin sheath because it can't take place there, and it will jump to the next node of Ranvier. This process is called saltatory conduction. As a result, the action potential passes much quicker along a myelinated neuron compared to an unmyelinated neuron. So if you just imagine being able to skip, 5 meters in a race whilst everyone else has to actually run those 5 meters, you're obviously going to get to the end of the race much quicker than them. So we can say that the action potential does travel much quicker along a myelinated neuron than an unmyelinated neuron.